Today we're going to talk about meiosis and genetic variation, and the goal of this video is to describe events during meiosis like independent assortment, crossing over, and nondisjunction, and explain how these events contribute to genetic variation. So what is independent assortment? The law of independent assortment states that pairs of alleles separate independently during meiosis. And this means that an allele on one chromosome has an equal chance of being paired with any allele on another chromosome. This happens because the way that homologous pairs of chromosomes line up during metaphase is random. So look at this picture. At the top, you see two identical cells. The way that the chromosomes inside those cells line up is random. So the red chromosomes here could line up, both of the reds could be on the right side, or you could have one on the right and one on the left. Those are the two possibilities. It's random how they line up. Now, this matters because by the end of this process, by the end of meiosis, the gametes that get produced, they can have different combinations of alleles in them. So you can see down here, one possibility is that the chromosomes have the little r and little y allele. Another possibility is that the chromosomes have a big R and a big Y. Another possibility is that they have little r, big Y, and another possibility is they're big R, little y. So those are the four possible combinations of alleles. And this variation is due just to independent assortment. Okay, let's talk about a second way that variation is produced during meiosis. This is through crossing over. So what is crossing over? Crossing over is the exchange of sections of genetic material between non-sister chromatids during prophase 1 of meiosis. So you can see here I have one chromosome. It's all in green. The left side is a sister chromatid and the right side is a sister chromatid. The purple chromosome here, it also is made up of two sister chromatids. And what happens during crossing over is non-sister chromatids swap sections of their chromosome. And you call the new product the recombined chromatids. So crossing over results in the recombination of alleles. And this produces even more variation in the offspring than independent assortment alone. So you can see down here at the bottom, these would be 4 gametes produced through the process of meiosis, but because of crossing over, you can see each one of these gametes produced looks different than the other, and this is where variety in or variation in the offspring comes from. So notice, although 2 gametes have the same combination as the parents, so you can see this purple one would be like up here, and this fully green and fully purple one would be like the parents up here. Although 2 gametes have the same combination as the parents, 2 of them now have new combinations of alleles, and that's what you see in the middle here. All right, last, we're going to talk about how meiosis produces variation through an event called nondisjunction. So what is nondisjunction? Nondisjunction is the failure of chromosomes or chromatids to separate during anaphase 1 or anaphase 2 of meiosis. And this results in a gamete that contains an extra chromosome and a gamete that has a missing chromosome. The gamete with an extra chromosome, we call N+1, and the gamete with a missing chromosome we call N-1. So just to show you what's happening, in this column, we have a normal division. So you have 2 homologous pairs of chromosomes here. You have a pair at the top, a pair at the bottom here. They line up in the middle, they separate, line up in the middle, separate. So notice all 4 gametes end up with 2 chromosomes inside each of them. But notice if non-disjunction happens during meiosis 1, so notice this homologous pair did not separate, then when this cell divides, it has an extra pair of chromosomes in it. And when that cell divides, these 2 gametes will have extra chromosomes in them. This one will have a missing chromosome. Same sort of thing happens during meiosis 2 if non-disjunction occurs. So, but instead of having instead of having 2 gametes with extra chromosomes, this time there'd only be 1 gamete with an extra chromosome in it and 1 gamete with a missing chromosome. Now, if one of these gametes is involved in fertilization, then the individual that results will have a chromosomal disorder. And if you've heard of Down syndrome, individuals with Down syndrome, they have an extra copy of chromosome number 21. And this would occur as a result of non-disjunction giving one of the gametes, either the sperm or the egg, an extra chromosome. And that sperm or egg was involved in fertilization, and that's how that individual got the extra chromosome. So to sum up, you've seen how you can get lots of variation through 3 types of events during meiosis: independent assortment, crossing over, and non-disjunction.